Hello, welcome to the MDC Connects webinar series. I'm Sarah Brockbank. I'm a lead scientist at Medicine's Discovery Catapult, and I'm hosting the session with my colleague Dan Bond. This is now the final webinar in a series of weekly webinars we've run weekly through the COVID lockdown period. So as the 9th session today in this series of 9 informative drug discovery sessions, each has been delivered by experts from the Medicine's Discovery Catapult and experts from our CRO network partners. The sessions are focused on aspects of preclinical drug discovery, and we started with the selection of the right target and then identifying the hit. We worked through optimizing the compound, and we had a focus on the chemistry involved. We introduced PK/PD and how that relates and informs efficacy models. And we're finishing the series by defining safety, which was the subject of last week's talk, and biomarker strategies today. So in this session, we're thinking about defining a biomarker strategy, and I'd like to welcome 3 speakers. Firstly, Gayle Marshall, lead scientist from here at Medicine's Discovery Catapult. Gayle is going to set out the importance of thinking about biomarkers early in your drug discovery project and defining a clear strategy. Then Helen, who's biosamples lead, also here at Medicine's Discovery Catapult, and she is going to tell us how to access the clinical samples required. And finally, Russell Garland, who's a group leader for analytical services at Charles River, and he will talk about biomarkers in the context of immunology and provide examples of how biomarkers can be identified and measured using different platforms, and what does validation of a biomarker look like. So we will take questions at the end of each presentation. If you wish to ask a question, please type these into the Q&A box, which you will see at the bottom of your screen, not into the chat box. And if you put your questions into the Q&A box, our speakers will pick them up at the end of their talk and address them then. So over to you, Gayle.
Many thanks, Sarah. Hopefully you can all see my screen and my slides. Just want to thank you very much for inviting me today to talk around developing a biomarker strategy. What I'd like to do is take you through what a biomarker strategy actually is and why you should have one, how you go about developing a strategy for drug discovery, and a couple of case studies where actually having a biomarker strategy has actually changed the direction of the drug in the clinic. So the first one is, why do we need to develop a biomarker strategy? As many of us know, the drug discovery pipeline, which moves from target identification to validation, moving into lead identification and optimization, and into preclinical. And then the idea is then at that point, if the drug has been validated to that point and is ready for clinical submission, you need to have a plan. Where's that drug going once it gets into the clinic, and who are you going to treat with that drug? Many drugs fail in the clinic, and it's cost many, many years to develop through this pipeline, and very expensive. And so whilst many compounds actually do fail in the preclinical phase, a lot of them still go into the clinic and actually fail there. And it's a very, very low percentage that actually get all the way through to registration and treating patient disease. So there are many reasons why these clinical trials can actually fail. Through things like lack of efficacy, so you're actually not having a response that you're expecting in the clinic. Things like pharmacokinetics and pharmacodynamics are just not at the right level, so maybe the drug's being cleared too quickly, or just not having a high enough amount of drug that can get in there to actually have the effect you need. It can have some off-target effects, so the safety profile just isn't right, but it's also the wrong strategy. So if you're actually treating the wrong patients with that drug, you will find that the drug will fail in the clinic. So the key is actually to understand what the key clinical questions are, build that testable and scientific evidence to show then that you can move from the preclinical into the clinical, and you've got some evidence already that you know that drug potentially has a way of working. And you can use these early studies to test those hypotheses that you can build in that early phase. So what is a biomarker strategy? So the strategy actually is to set out the set of clinical questions you require to go into the clinic. So the first one's usually quite easy. So what disease is your drug going to treat? A drug that would work in oncology would not work potentially in autoimmune diseases, for instance, but there are many, many other questions that you might want to know. So you might say, okay, I know what disease, but then which patients within that disease, because often there are so many different patients which have a very different disease profile that require to be met before you can actually identify which ones are going to work with your drug. Then at what dose are you going to give those patients that drug? And then how often? So is it going to be a daily tablet or a monthly injection? Those kind of things need to be worked through very early on. One of the big questions is actually, is your drug going to be better than what's already in the clinic? So the standard of care that patients are already receiving, many drugs will fail because they work, but actually they're no better than what's already available to patients. And so that has to be thought through before even going into the clinic. Often combinations are a really good option because you have then the chance to double your chance of success. So in theory, that if you have already, say, the standard of care, if you were to combine with your new drug, then you have the opportunity to improve that standard of care treatment. And then thinking about things like acquired resistance. So if you were to treat a patient long-term with a drug, how do you know that that patient's then not going to acquire a resistance to that drug? And if that's the case, what's the mechanism? Is there a way to develop another drug that you could use as a backup once those patients have relapsed their disease? So by having a strategy and understanding those key questions, you're able to go back into the preclinical setting and work through how you can work through those questions that then you can test that in the clinic to mitigate some of those risks, and actually then have a very robust clinical strategy moving forward. So when do you think about introducing biomarkers? I'm a bit of a stuck record on this one, I'm afraid. So biomarkers, for me, has to be introduced right at the beginning of your drug discovery pipeline. So whilst you're thinking of target selection and working through the mode of action of your target with your inhibitor or activator, what you need to know are all the markers that are involved in that. And what you can do is look at a lot of multi-analyte assays that allow you to measure many different analytes. So things like proteomics and genomics, using things like imaging technologies to really actually understand the disease that you're aiming to target, but also the target that you're going in with for your drug, to understand exactly what's going to happen so that when you actually get to the clinic, you have then a handful of markers that you might want to monitor moving forward. As you progress through your development of your drug, you have the ability to build really robust assays to allow you to test that. So whilst you're testing it in the preclinical setting, you can also be thinking about accessing clinical samples to see whether those targets and how variable they are within the disease. So whilst you're reducing the number of analytes as you progress through your validation of your biomarker strategy, you would then look to increase the number of samples that you would then test that biomarker on, and hope that by the time you get into the clinic, you have a very well validated and robust clinical assay. So these are some of the key questions that can be built into a clinical trial design. So things like pharmacodynamic markers. So how do you know that your drug is actually hitting your target? So once you have treated a patient and that has the disease, this data that you can build through your pharmacodynamic readout will help you set things like your dose and your schedule of that drug. It could also provide your proof of mechanism and also give you some go, no-go decisions. So can you go up to the top dose, or do you actually need to? Could you go in at a half dose because you know that you're already wiping out your target at that point? Markers like proof of principle are things like proliferation markers, for example, or cell death markers. So do you have a downstream effect by hitting that target? Are you able to modulate the morphology of your disease? And then things like proof of concept. What are the clinical effects? Is your patient actually responding to that drug? We measure things like predictive biomarkers, can you actually predict you actually will have an effect on that target? But also patient selection, which is the most common biomarker that's measured in the clinic. Which patients will be best to respond? So which patients, if you were going to pick out a patient that was going to actually work on, what's that subset that you need to select to make sure you have the best chance of success for your drug? There are many safety biomarkers, and I think there's already been a safety set of presentations. So I'm not going to go too much into safety, but safety biomarkers are also quite key to your biomarker strategy and what considerations you actually need to make sure you're mitigating against. Also, as I mentioned before, resistance. What are your potential markers of resistance that you could be monitoring through your drug discovery approach? So that means then you can see whether a patient is going to become resistant before they actually start showing the effects of resistance. And then what are combination options? There could be multiple options that you could combine with your drug to actually give you a much better effect. And then, like I mentioned before, how does it compare to that current standard of care? And that is actually quite a key question. So it sounds quite easy, many questions to ask, but actually, even though biomarkers is such a broad word that people use, there are still very, very few biomarkers that are being used in the clinic. And it was noted back in last year, even 880,000 papers in PubMed directly related to biomarkers. And even though laboratory technology is advancing over time, we're actually finding that there's still only a handful, really, of clinically relevant biomarkers that are in use. So maybe it's not quite so easy as we think. So biomarker uptake are actually have problems having that lack of clinical utility. So the marker itself is very complex in some cases and can be underestimated. So when you're actually looking to monitor a marker, how do you know at what point you should be taking samples to then check to see the levels of that marker? The lack of understanding of the pathology and the heterogeneity of the disease. So if you're taking, for instance, a biopsy of a tumour, how do you know that your marker is equally distributed across the whole tumour, and that by taking that biopsy, you're actually being able to take the subset of cells that you're looking for? Also, the use of inappropriate samples for discovery and validation. Now, this is quite key, and I know Helen's going to be talking a little bit about how to access samples, but the key part of that is actually making sure they're the right samples, because you can look at building a biomarker validation set, but actually if it's based on the wrong set of samples in the first place, then the biomarker might not translate into the clinic. And then methodology limitations. You need to know what the how robust the assay, but also the variability of that marker and how quantitative you can read it. So I've got a couple of oncology examples here. So many people will start with the expression of the target. So if you're going to give a treatment in oncology to often inhibit oncology-related targets within disease, the first thing you potentially would look at is how much of that target is expressed within the tumour. So we have a couple of cases here where drugs like Herceptin. So in breast cancer, patients who receive Herceptin are tested for HER2 levels, and if they are incredibly high using an IHC assay, those patients will then receive Herceptin. All the patients who have moderate to low levels of HER2 will not get that drug, because they know that they're not going to respond to the antibody. There are a whole range of monoclonal antibodies that are approved now for immunotherapy treatments, and they are also ones where the target amount is predictive. So by high levels of these targets actually means that your chances are higher of responding to those drugs. Then we have a group of compounds that actually the fact that the target is actually there is predictive enough. So you have things like the antibody crizotinib, which acts on ALK1 and ROS1. And this is already approved now for non-small cell lung cancer. And it's found that actually it's the fusion of the ALK1 that predicts response. There are other drugs there as listed, but it's not just the protein itself that is the responding factor for these drugs. It's the fact that it's the form in which those biomarkers have to be detected to say whether those drugs are going to work. And then we have drugs where actually, even if the target's there, and even regardless of the level of those targets, they're still not predicted to those drugs being responders. So things like Lynparza and cetuximab, it's actually potentially, well, it is actually mutations that are detected within those patients actually say whether they're going to respond or not. So a quick example here is of Iressa, which its generic name is gefitinib, and it inhibits the EGFR epidermal growth factor receptor. And it was launched back in 2002 in Japan, was its first place to be launched. And 60% of lung cancer patients actually express EGFR. So the obvious thing to do is to actually look to say, let's recruit patients who have high levels of EGFR receptor, they're going to respond to this drug. But actually it was found that that was not the case, and many trials failed in phase 3 because the levels of receptor did not equate to whether a patient was going to respond or not. Then in 2004, it emerged that actually by sequencing the tyrosine kinase part of the receptor, that mutations were identified, and actually activating mutations, which was the driver for the lung cancer disease. And if you could then select patients based on that mutation, actually you had a much greater chance of seeing that Iressa or gefitinib was actually working in those patients. And so now gefitinib is actually approved as first line in EGFR mutated advanced patients across 85 countries. On top of that, though, only half of those patients still respond. So there's still 50% of patients that still have that mutation that don't respond. And it was also found then they had a second mutation, which was like a gatekeeper mutation. And that gatekeeper mutation was stopping Iressa from working. And so then another line of therapy has been developed to actually target specifically that gatekeeper mutation. And that drug approach has actually already had a patient approach right from the beginning, which has made it progress through the drug discovery pipeline very quickly. So this is a really great example where actually by having a really clear defined biomarker approach allows that drug to then be targeted to the right patients. So lessons that are learned is to really understand the biology, to carry out those preclinical translational science experiments, and always to look at the tissue. So again, samples are everything. So if you don't have samples, there is no way you can test a biomarker. And actually, by making sure you have really good quality samples allows you to really give that biomarker a chance to do that predictivity. So it does matter what you measure, how you measure it, and define your positive cutoff. So things like Herceptin, as I mentioned before, you need high levels of Herceptin to work, but actually there is a cutoff point. So how do you define that cutoff to say you have to be above this level to be able to respond? So within the medicines discovery catapult, we do have a translational science approach where we can build some of that deeper understanding of the disease and the drugs that have an impact on that disease using multimodal technologies. So we can look at things like transcriptomics, proteomics, metabolomics, and actually really understand the way that the disease is and how drugs have an impact on that. And we look at that in clinical tissue. So we look at frozen FFPE tissue and also circulating markers in blood. And that allows you then to help organizations much better design that clinical study, give them evidence in the preclinical setting from clinical tissue that allows them to find what their PD markers should be, what their predictive biomarkers potentially could be, and we can retrospectively test for those, as well as other markers that actually we should be monitoring whilst you're doing your drug development, but we can then see what that data looks like once you go into the clinic. So this is just a quick slide on some of the technologies that we have within the catapult to allow us to do that. And I know Russell's actually going to give more of an overview of technologies that you can look at to do your biomarker discovery and assay build for analysis in the clinic, but focusing on things like transcriptomics, metabolomics, lipidomics, proteomics, all the omics basically, but then also zooming in much more on where those biomarkers are actually being expressed, because we often find that many diseases are heterogeneous, and so you do need to know exactly where they're being expressed, whether your drug can actually penetrate into the tissue to access those targets so that they can have an impact. And all of that can influence then your approach in the clinic. So there are 4 key areas actually developing an assay in the clinic. And these are absolutely crucial to making sure that assay is measuring what you think it should be measuring. So you need to know that the biomarkers are relevant to what you're looking at. So understanding that biology is critical. Making sure the assay is suitable. You need to know that the assay is robust, that you don't need a huge amount of sample to be able to measure that marker, that you can actually run it again and again and again, and you will see the same data every time. And that will allow you to start building up what those go, no-go thresholds start to look like. Making sure your study design is key.
Sorry, do you want to speed through a little now?
Oh yeah, sure, sorry. And making sure that the samples that you can receive are able to happen in the real world. I'm just going to click through these. So just through to the last slide. Sorry, I've overrun my time. But just to say that to have a clinical biomarker strategy and being able to use it in the real world, it is key to actually know what your questions are, to make sure your assay is suitable, and make sure your study design is key and that samples are available to allow you to do that. So just a couple of acknowledgements, and I'd like to take any questions if there are any.
If you want to just take it and choose
and do that offline, absolutely.
And then answer by text.
Okay, that's great. Thanks, I'll do that.
Okay, thanks, Gayle. I'm Helen, the biosamples lead at Medicines Discovery Catapult. I'm just going to talk through some of the challenges of accessing clinical samples with particular relevance to the SME life sciences sector. Next slide, please. So just to put this into context of why MDC has a sample access theme, several years ago when the organisation was setting up and defining areas of priority, a report was produced called State of the Discovery Nation, which surveyed UK SMEs around the various challenges in R&D. And one message that came back pretty strongly was that while access to clinical samples was considered to be very important, in practice this was pretty challenging to achieve. So I'm just going to talk through a few factors around why SMEs need samples in the first place, where they might get them from, and what they do with them. Some of the barriers to sample access within academic biobanks and how they might be overcome, including how industry could help to streamline the process. Who needs to act and get involved in both highlighting and solving these issues, and why it's important to use UK bioresources. Next slide. So thinking first about why SMEs need samples. Well, it's part of their core R&D activity. They need samples in order to progress their assets, so developmental drugs or diagnostic products, and to be able to launch these new products in order to deliver patient benefit. There are a number of places that SMEs can look to access samples from. They could set up collaborations with clinical academics or other key opinion leaders where they have those contacts. They could look to get them from NHS or academic biobanks, and many of these can be identified through the UK CRC tissue directory. They could get them from commercial sample supplier organisations, a number of which are based in the UK, and these typically act as virtual biobanks and can acquire samples from global sources. Or they could use other procurement platforms that deal with a full range of products and services, as well as access to samples. And the samples can be used in various different ways, for things like method development or assay validation, sequencing. It's part of a regulatory submission package, as Gayle's already talked through, for biomarker analysis, and also used to do things like compare normal and diseased tissue states or comparisons across matched samples, so typically blood or PBMCs and tumour sample from the same patient. Next slide. So barriers to sample access within the UK, and the first thing to say is the problem is not the absolute lack of patient samples in itself, it's rather the number of them that are accessed for use. In some cases, biobanks are not adequately promoted and can be hard to find if they don't have a dedicated website or a listing on the tissue directory. Researchers may not be aware of individual biobanks, particularly in very small early stage organisations where there could be limited experience in sample acquisition. And this is different to large pharma organisations who can have whole teams of biobanking experts who work on sample access. There's a national infrastructure to some extent through UK Biobank and Genomics England, but these tend to prioritise the larger pharma organisations rather than SMEs. And because there's no UK-wide infrastructure, there can be issues encountered with things like individual access committees, so the timeliness of when they do the reviews, different requirements of different committees, the governance requirements, so things like ethics approvals, confirming material transfer agreements and agreeing terms, and also the speed of access, which is really critical for SMEs as they need to move quickly to progress to the next milestone or investment decision, otherwise they struggle to survive. Next slide, please. So thinking about how industry could help the process in making samples available to them. I mean, the first thing I would say is to have an understanding that even prior to the COVID-19 pandemic, biobanks were under unprecedented service pressures to deliver diagnostic work, which has only escalated in recent months, and the resources are extremely stretched. To ensure that requests are submitted with as much lead time as possible, to think about ensuring that the request has been scoped out to the extent that it's fixed and it isn't going to change. So for example, if you've put in a request for fresh frozen tissue and then you suddenly switch that to fresh tissue, that is massively going to impact on your timelines. Identifying the essential criteria for the request from the nice to haves. So often see this with the amount of data that researchers request for the sample, and just a plea to ensure that if you're requesting a large amount of data, that you do need it all and it will all be used. Next slide, please. So then, overcoming the barriers from the biobank perspective, there are a number of things that they can do to overcome some of the issues or to consider in order to exist as a sustainable service. The first will be to define a robust cost recovery model to include all aspects of the end-to-end process required. So from consent and taking the patient sample through to making that sample available to a researcher. And this model obviously varies according to the different operational processes that occur between institutions. To maintain customer awareness, so how findable they are they listed on the tissue directory? Do they have a dedicated and user-friendly website, or are they embedded within a trust or a university website and could be hard to discover? Customer management is important, so industry typically require a prompt answer, and it's important to get an early read on whether the request is feasible or not. So even if the answer that comes back is no, it's important that they hear that answer quickly so they know to look elsewhere. And thinking about operational processes, is it clear how you apply for access? Can you download the request forms on the website? How is the access committee set up and how does it function? So the frequency of meetings, is there any patient and public involvement representation on there? And are there clear timelines set for review? Documentation, I think about whether there are things like material transfer agreements available on the website. Are the terms of that agreement negotiable? For example, industry will find it difficult to sign off on these agreements if things like the IP clauses are too restrictive, if it impacts on their proprietary tech. On delivery resources, so does the biobank have dedicated staff to manage the end-to-end process? Is it managed by like an operations manager and a technician? And who actually manages those requests? Who has the authority to progress those? Does it fall back to an operational manager, or do you have to defer to the PI every time? And then also patient engagement and feeding back the results at the end of projects maintains patient engagement and supports future research taking place. Next slide. So thinking about who needs to act to overcome some of these challenges. So within the UK, we have the Health Research Authority, or HRA, who have an agenda around transparency in research and conducting research for patient benefit. And currently, registration on the UK CRC tissue directory is a condition of approval for a research tissue bank, but not all of them do actually do this in practice. HRA maintain it's not their role to enforce. They get involved in cases more like where there's a serious breach of patient consent, but they feel that it's not their place to try and enforce that any further. Then there's the Human Tissue Authority, or HTA, who would like to see a greater proportion of banked research samples used for valid purposes. But their remit involves other areas such as transplantation, post-mortem, public display and anatomy. Research actually makes up a small proportion of what they oversee. And last but by no means least, the patient groups. Where patients donate research samples, they do have an expectation that they will be used to their maximum capacity. And when they find out that the typical use rates of a biobank can be 15% or lower, they're understandably disappointed with this, and it does raise some clear ethical issues. And then why it's important to use UK biobanks? Well, two main reasons. Many of the biobanks are set up with grant funding for the first few years and will need to self-sustain after that period of time. A number of biobanks within the UK have had to close or merge with other bioresources, which creates more issues with the samples that they hold and the sample collections that they're able to supply. And if they're self-sustaining, they deliver benefit to UK PLC by being a valuable resource. So that was about the end of what I wanted to say. So if there are any questions, take those. So I just have one question, which is, what do you expect the future requirement of clinical samples to be from the SME sector? So I'd expect this to only increase further, to be honest, particularly within the diagnostic companies. Thinking about the new IVD regulations coming in by 2022 means that some products which are not currently regulated will need to be brought under those regulations. So I think that will be one area where we will see quite an increase on where samples will be required. So if there's no further questions, can I hand over to Russell Garland of Charles River to talk further about biomarker identification?
Okay, thank you very much, Helen. So yes, my name is Russell Garland. I work for Charles River Laboratories. I'd like to thank the organisers for this opportunity to speak in this biomarker section of the MDC Connects webinar series. And I'll be focusing today on assessing assays of immune function. So I'd like to step back first and just remind ourselves what's the definition of a biomarker. It can be anything that's measured to give us an indication of the normal biology of a process, or normal physiology, or of disease pathology, or of a response to therapeutic intervention. And there are several different categories of biomarkers listed there, several of which Gayle has spoken about previously, and some of which I'll elaborate on further later on. So I'm sure you're familiar with this type of schematic diagram of the drug discovery pathway, from target discovery on the left through to clinical development on the right. Now we can exploit a number of primary human cell-based assays to assess the mechanism of action of a compound, and we can also support in vivo efficacy models. What we've also come to realize that is if we use some of these primary cell-based assays but uplift them to a higher level of validation, we can use them as exploratory biomarkers to support early phase clinical phases of the project. Now, immunology touches a number of different therapeutic areas. We may be looking to boost an immune response in the context of an infectious disease or in the context of immune oncology, or we may be wishing to reduce an undesirable immune response in the context of an autoimmune condition. And I'll be describing some platforms that can be applied across these disease indications. So here are some examples of the techniques that we run. You may be looking for a protein product stimulated by immune response, such as an antigen-specific antibody titer or a cytokine produced in response to a stimulus. And typically we might use ELISA or multiplex Luminex technology in these kind of examples. Quite often, if you measure normal serum or plasma, there may not be very much to measure, so you might need to induce a response as part of the process. You may want to look at the message for a particular biomarker by qPCR or NanoString to see whether a particular biomarker has been modulated by stimulation or the disease test compound. Or you may be looking at an increasing frequency of antigen-specific T cells by ELISpot, for example. And flow cytometry is another extremely powerful technique we can use to look at the frequency of cells responding to a particular antigen or in any particular sort of subsets of T cells, for example, as well as just a normal immunophenotyping type analysis. So I'll be elaborating on some examples of these. So in the preclinical setting, I'd like to start off by picking an example of NanoString technology. So this is a fully automated system where we can measure the expression of up to 800 genes simultaneously. So the cartoon on the right-hand side describes a target RNA molecule, which is then bound by a capture probe. The probe is biotinylated, which allows the complex to be immobilized on the surface of a cartridge. And there's also a reporter probe, which is shown there as a kind of multicolor barcode, if you like, and then a high-resolution CCD camera takes an image to allow quantification of each target RNA species. So this, with relatively little hands-on time and relatively little bioinformatics, you can measure a broad range of genes within an inflammatory biomarker panel. So in this example, what we've done is we've taken whole blood and we've stimulated with LPS, which is a well-known stimulus of the innate immune response through toll-like receptors. And what we've done is we've stimulated in the presence or absence of dexamethasone, which is very topical because it splashed all across the national news in the UK yesterday as being a cheap steroid anti-inflammatory, which has been shown to be suitable for use in severely ill COVID patients. But in the context of this in vitro assay, we're stimulating blood with LPS with or without this inhibitory agent. And after an incubation period, we're taking the supernatants and looking at secreted proteins by Luminex, which I'll describe on the next slide. And the RNA is extracted from the cell fraction and analyzed using qPCR and NanoString. So the graphs on this slide are volcano plots with a kind of characteristic V shape. So for any given biomarker or analyte, it's plotted in the graph with an upregulated gene being shown on the right of the plot, and downregulated genes are shifted towards the left. And the higher up the y-axis a point falls, it means it's a higher significance of change. So on the left-hand plot, those genes within the red circle are those which have been highly upregulated in response to the stimulus. Now on the right-hand side, we're looking at the comparison of LPS with or without the dexamethasone, showing that those genes to the left-hand side in the blue circle are downregulated by the test treatment. So this is an example of a technology where we can measure multiple different genes and profile them at the same time, and I'll use these to select our biomarkers and follow up with a higher throughput technique to then validate. So this slide shows samples from the same experiment where we're looking at by qPCR at the message or by Luminex at the protein on the right hand side, and we can show that by base techniques we've got a large window of effect between the stimulus and the unstimulated control with the LPS, and that these four inflammatory cytokine markers are all reduced by the presence of the dexamethasone. So the two techniques show good concordance. So that's an example of using a broad kind of screening technique and then focusing on a smaller panel of analytes. So then when we come onto the clinic, as Gayle described in her talk, there's multiple different categories of biomarkers. So for example, we heard in previous weeks in this series how PK is what the body does to a drug and PD is what the drug does to the body. So in a PD biomarker, we're looking to measure some kind of biological response which has occurred due to an engagement of the target by the medicinal product. So we're looking here at proof of mechanism. typically in an early phase clinical trial, this will be an exploratory biomarker to support the sponsor's go, no-go decision. So patients won't actually be treated on the basis of the result that we would generate with these analyses. And it's PD biomarkers I'll be describing today. So other categories of biomarkers used at these stages include predictive biomarkers, where you may be selecting a subset of patients to be suitable for a particular treatment, and safety biomarkers, which typically need to be performed to a higher level of validation because they're often the primary or secondary endpoint of the clinical trial. So then it is possible to perform clinical sample analysis. And the first thing we tend to look at is nailing down within the kind of four walls of our laboratory, as it were, how our operators perform at our machines with the batch of reagents we are going to use for this trial. However, you also need to think about upstream factors, so sample logistics. So how is the sample taken, what anticoagulant is used, and is there any sample shipping, storage, or handling involved? So you need to kind of, during your validation phase, you need to think through that process and design a fit for purpose validation to control for the major factors. So typically what we would need to do here would be to collaborate with a clinical center and contribute to the laboratory manual to describe how the samples need to be handled, shipped and stored for our analyses. So assay validation is important. So here's an example of a flow cytometry test where we're looking at cell surface expression of several markers on immune cells in EDTA anticoagulated blood on the left. However, when we try to fix these samples by transferring the blood into a fixative instead, to try and stabilize the marker to allow fixation, so samples could be batched or shipped from a more geographically distant site. What we found was we lost expression of some of the markers that we were interested in. So we needed to work around the problem of the epitopes or the antibody clones themselves not surviving the signal, not surviving the fixation process. So during the validation, you need to mimic as closely as possible what the trial samples are going to be exposed to. So I'd now like to move on to an example of an ex vivo whole blood stimulation assay, where the readout ultimately is quantification using Luminex. But again, we needed to think through the process from blood collection from the trial subject through to any shipment and delay in the draw to processing time. There was an activation phase in the assays I'm about to describe. So we needed to store samples at a suitable time point. And we also needed to confirm that we could recover those cells, those analytes rather, and that they were stable upon storage. And then for the Luminex itself, we needed to look at an informative assay panel, confirm analyte precision and how the standard curve performed. So this is an example of some of the assay validation steps. So the data doesn't tend to be typically exciting. It's typically lots of tables showing how the standard curve performed, and also intra and inter assay precision, where you're looking for good variability. But this is the kind of assay data you need to tell you how your assay is performing and whether you can batch the samples. So again, this slide lists some of the characteristics we may choose to look at during the validation phase. And on the upper right-hand figure, we're looking at superantigen stimulation with SEB and showing that we've got a robust window of response between our unstimulated control and the SEB stimulated, but we do see some variation between donors in terms of the magnitude of response. What the lower figure shows is that if we spike standard recombinant cytokine standard into plasma and then measure these levels after 1 or 4 weeks at -80, that we can still detect the analytes after a prolonged storage. So this would then facilitate batching. This is then an example of a biomarker analysis we used, an exploratory biomarker we used to measure inflammatory cytokines, and in this case IL-6, in patients, a cohort of patients with an inflammatory skin condition. So we took, this being a PD biomarker, we took samples prior to and following a 28-day treatment period, and we were then able to compare the baseline with the post-treatment response. We were blinded in this example, but the sponsor then unblinded and produced this waterfall plot showing that compared to the placebo, there's a pronounced downward shift in the subjects treated with therapeutic in their responses to IL-6 in this example, thus suggesting this was supportive of their test treatment having potential to treat inflammatory diseases. now we come on to another autoimmune example. We were involved in a multi-centre EU-funded study where we were taking PBMCs, or peripheral blood mononuclear cells, from patients before and post-treatment. And then we were shipping these samples to our laboratory, and we performed a number of analyses, so qPCR, Luminex for inflammatory cytokines, and some some panels using flow cytometry, which is the subject of what I'll be describing further here. So we had a Treg panel, but also a T effector panel. So the workflow here was that the frozen PBMCs from patients prior to and following treatment were thawed and then established in culture in the presence of the therapeutic peptide or a positive control antigen. And the gating strategy shows that we were looking at CD4 cells for the proportion of these that were proliferating, as evidenced by their Ki67 expression. And then we were furthermore looking at these to see what inflammatory cytokines they were producing, hence the frequency of cytokine producing cells to the antigen of interest. What we would typically then do is to compare the expression in the pre and post-treatment samples to see whether the therapeutic had modulated the desired effect. So why go to all this trouble? Well, it's essentially, clinical trials are an expensive business, and as the phase of the trial progresses and the number of subjects increases, so the cost goes up. There's a particularly high attrition rate at phase 2, predominantly due to a lack of clinical efficacy, some of which may be due to poor target engagement. So what we're showing here is some data from the AZ pipeline on the right-hand side, showing that programs that have a PD biomarker associated with them, so target engagement or proof of mechanism is demonstrated, these have a positive impact on project success. So projects with a demonstrated proof of mechanism have got a higher chance of progressing to phase 2, phase 3 or launch, whereas you want to avoid the situation where you reach phase 3 on a clinical trial, but you've lost faith in the target, or you haven't shown proof of mechanism. So we think that adding PD biomarkers can have a positive impact on project success and add value. So then, by way of a summary, in some respects this reiterates what Gayle mentioned in her earlier session, which is if we can include response biomarkers early in the preclinical stages of the drug discovery process, we can confirm target engagement. If we can select informative biomarkers, these can also be translated to the clinical phases. And whilst early phase first in human trials are often associated with the primary and secondary endpoints, rather, are mainly associated with safety, if we can build in some additional exploratory biomarkers at this stage, we can add value and provide proof of mechanism to support the sponsor's go, no-go decision. So the approaches I've described are possible and can be applied to clinical trials, but they need to be designed around the particular needs and the particular logistics of a given trial. And in this regard, validation is key. So thank you very much. And I think I will have time for a few questions. So one is, what's the typical cost and time frame of developing a biomarker? Well, essentially the cost will be to some extent driven by what platform is we're looking at and how many biomarkers we're trying to measure. In terms of the time frame, I think I would typically say that if we're already speaking to clients at the preclinical phase and we understand what the program is, it does make it easier to translate assays into the clinic. But typically, even if a proposal was signed today, we would need at least 3 months or so to get a suitable batch of reagents in and to confirm the assay is fit for purpose. So obviously, the closer what we're measuring is to something we've already done, we can save some time on that. But again, it depends on the complexity. One thing you can speed up is time. So if you're looking to ship samples from a geographically distinct or distant site, we obviously need to think through logistics and any solutions around that, such as sample fixation, or it may be that we need to advise the clinical site on some sample processing and storage that can happen there, so they can be shipped later at a later date in the frozen state. but yeah, it's kind of the one thing I would stress is that, you know, we shouldn't scrimp on validation. We need to expose the samples during the validation phase to the similar conditions as the clinical samples are going to be exposed to, because obviously we're going to be producing a result at the end of the day that the client needs to have confidence in. Another question is around what housekeeping genes or sample comparisons are used for the NanoString. So essentially, we would always be comparing a pre and post treatment sample from the same patient. So there's a kind of pairwise comparison, kind of pre and before and after treatment. There are different panels, some of which have got 800 genes in, others of which have got, say, 200 genes, or it depends on what you're trying to get out of the analysis, really. There's a lot of different control steps in there to make sure that the samples are in a good state and good enough integrity, or the RNAs are good enough integrity to get to good signal. It's very much a case of making sure your results pass all the quality control steps to have confidence in the data you've generated. And finally, we've got a question around ELISA and looking at analyte detection and the fact that this can differ between kits, technology and manufacturer, and to what extent would I agree with this statement, and what would any recommendations be? I think it's always difficult if you're trying to follow a particular kit or a particular paper. It's always difficult trying to chase the numbers which has been generated by someone else. But I think if there are some quite commonly used manufacturers in the context of ELISA and Luminex, for example, and if you, in the first instance, if you've got a trusted relationship with the supplier, then quite often they will have done some validation and the specificity and sensitivity they would expect with their kits. So it's usually the best first port of call would be to go to some suppliers you know. If it's a particularly odd analyte that you can't find, then you may need to reach out and check with the provider what validation they've already done and kind of test it in-house yourself. But there's not really any substitute for, once you've got that reagent in your hands, it's kind of showing that it's fit for purpose in your hands with your operators and under the conditions that are going to be used for your particular preclinical or clinical phase.
Are you happy you've answered the questions, Russell, or is there any others that you want to answer?
I think those are the ones that are the easiest to take on at the moment. Yeah, thank you.
Okay. Hi there, it's Sarah here, just finishing us off for today. So thanks to Gayle and Helen and Russell for steering us through the last in this series of MDC Connects webinars with the 3 talks today, which demonstrated, I think, quite clearly the importance of defining your biomarker strategy early in your drug discovery programme and then some practicalities of how to go about it. So finally, this has proved to be an extremely popular series of webinars, due in no small part to the expertise of the speakers and the quality of the talks that we've had. May I take this opportunity to thank all of the speakers who've contributed to this excellent drug discovery series, and also to Dan Bond, who's running the show behind the scenes. Thanks to all of you for listening and for your questions, and goodbye for this series. Thank you.